Good evening, and I hope you've had a very wonderful Monday. If you live here in the Houston area, you've had a cold Monday. We've had a little bit of interesting time here in Houston. Things are cold, and we had some snow, and it's a little bit exciting because just before the show, my oldest daughter, had a little skid in a car and totaled it. Fortunately, she and her friends are not hurt, and so I had a little bit of excitement starting. I hope your weekend was restful and productive, and of course, your day was productive. And for the Houston area, I imagine you may have had power outages or dealt with the snow, but hopefully you got through it, and we've gotten through it. But we have a very exciting show for you tonight. I'm excited about this topic because we're going to talk about the science behind plant-based nutrition. And I've been recommending this to my patients for nearly 2 decades, and we've seen amazing results. And our special guest tonight is going to help us go through some of the underlying mechanisms of why a plant-based diet works. So stay tuned for this exciting show. And if you like what you see, I want you to like, share, and subscribe. So let's get started. Okay, welcome back and happy President's Day. We've got a long show tonight, and so I want to get right into it. A special guest is a gentleman who's worked with us here at Montgomery Heart and Wellness back several years ago. It's Mr. Rami Najjar, and he is a PhD candidate at Georgia State University. Rami holds a master's degree in nutrition and food science at Texas Women's University here in Houston. He also has his bachelor's in nutrition and food science at the University of Houston. Rami's currently working on his PhD, and he's interested in the effects of plant-based nutrition and heart disease, specifically the effects of berries on heart failure. He and I have collaborated on a number of research studies, and we are actually working on an upcoming project. But what we're going to talk about tonight is the effects of plant-based nutrition on heart disease and also chronic illnesses in general. And he's going to help us understand some of the details of the biochemistry, what happens inside your cells. Rami has a very impressive resume here. He's had numerous honors and awards, research conference travel award, first place poster presentation at the University of Georgia State University in 2019, second place poster award in 2018. He also has the Carolyn and Matt Corey Endowment Scholarship, and he's made the Dean's List from 2011 through 2014 in Houston in his graduate program. His professional memberships in American Heart Association, American Society of Nutrition. He's a manuscript reviewer at BioCell, as well as The Lancet, and also an American Journal of Lifestyle Medicine reviewer, and a review of evidence-based complementary and alternative medicine. He's trained numerous students at the University of Georgia State University, in 2018 and 2019. So there's a pretty extensive teaching requirement there. Done a number of clinical research, basic science research, and the like. And so we look forward to bringing Rami on. He's a very hardworking graduate student, very knowledgeable, and I'm confident you're going to learn a lot tonight. And so without further ado, we're going to bring Rami on. Hello, Rami. How are you doing?
Hey, Dr. Montgomery. Thanks for having me.
Welcome to the show. Okay. Good to see you again. And I recognize that backdrop. You're in the lab, so you were working tonight, and that's a usual place for you. And I just want to let people know that, graduate students, they have a broad spectrum of responsibilities. And as I outlined in his bio, He has teaching responsibilities. He has research and publication responsibilities, and he's working in the lab. I mean, I called him late last night, and he was working on cell prep. So he's done a lot of biochemical experiments in the lab as part of his work. So it's pretty exciting work. Before I get into Rami's presentation, I'm going to bring on our regular panelist, Dr. Celeste Palmer. Celeste, how are you doing this evening?
I'm well.
So I can tell you have power. That wasn't the case earlier today. So as I share with everybody before coming on, we've been dealing with some issues here in Houston. I had power the whole time, but my oldest daughter totaled her car, and so I had to deal with that just before the show, Sam. And so she totaled her car, and then when I got back, the backflow pipe for my sprinkler system had burst. It was wide open. And so it took about 15 minutes for us to get it closed because it was part of the handle was frozen. So we got wet and all of that stuff. And so all of that happened before coming on. But anyway, a little excitement to get the adrenaline going. But what I want to do is share with the group the topics we're going to talk about. And as I said, we're going to go into the whole concept of the biochemistry of plant-based nutrition. So what I want to do is I want to share, this is going to be part one of a 3-part presentation. So Rami and I, we're going to break this down in bite-sized segments. And so part one, we're going to talk about inflammation and other biochemical effects of plant-based nutrition. And so Rami, why don't you get us started with this whole concept? I know you have a slide presentation. I'll bring that up here. And so inflammation, a lot of patients know about inflammation. We hear about inflammation. And when I explain it to my patients, I always give them the old, you fall off your bicycle and bruise your arm example analogy. And people tend to hold on to that. What should we know about inflammation? Because it's an important concept. And so why don't you get us started there?
Right. As you can see here, inflammation underlies essentially all chronic diseases. And the mechanisms are actually very similar when you look at the cellular level throughout all these chronic diseases. COPD, heart attack, liver disease, renal dysfunction, Alzheimer's, all kinds of diseases. Inflammation is essentially the root cause at the molecular level. I'm hoping to share some of those details as well to try to get an understanding.
And Rami, get closer to your mic somehow. Turn it up so we can hear you more clearly.
Is that better?
Yeah, that's much better. Or maybe, I don't know if you can turn it up or whatever.
Yeah, I just had get a little closer to the laptop here then.
Oh, perfect. No, that's good. Yeah.
Okay, good.
We just want to see a nice smiling face. That's all. So anyway, so you were going into inflammation in terms of the mechanism, how it's similar in different situations. Go ahead and continue with that.
Right. So when I was saying at the cellular level, all these chronic diseases have very, very similar pathways. It's all the same root cause leading to these chronic diseases. And my belief is genetics doesn't necessarily determine whether you'll get a chronic disease, but perhaps you're more predisposed to cardiovascular disease. So your genes say you'll get that. So, if you eat a poor diet, you could get heart disease. But if you're predisposed to perhaps diabetes, you'll have a poor diet and lead to diabetes. So inflammation is at the root cause of all these chronic diseases. And I'll get into some of those details shortly.
Rami, you hit a key point I want to just highlight there. So you mentioned genetics, and oftentimes people ask about genetics, and genetics will determine how you break down, not necessarily that you break down. So exactly. You have these 4 arrows here, and your genetics may determine which direction you go. But if you, as you said, eat a poor diet or have a poor lifestyle, you're going to have the underlying physiology or pathophysiology, if you will, or pathobiochemistry, if you will, that's going to make you sick. And so your genetics will determine how you get sick, not necessarily whether or not you get sick. So that I like that point that you made there, and I just want to highlight that for our listeners. But go ahead.
Right. It's so if we look over here, a single meal containing meats, so either fatty meats or lean meats, can increase inflammatory cytokines as well as CRP. So TNF alpha, IL-6, CRP, these are typical laboratory values. I know that you at least look at. CRP is widespread, but
Yep.
this is postprandial within the first 2 hours. So
Wow.
baseline 1-2 hours, you'll see a spike in inflammation. And yes, fatty meats, you'll get a greater spike, but even with lean meats, you'll still get an increase in inflammatory cytokines and proteins in the serum.
Yeah, and that's important for people to understand. And you know, I wish, I mean, maybe one day you and I will do this study, but you know, this is a full meal to after. I would like to do like one bite.
Yeah.
So,
Go ahead,
so
Celeste.
these are taking at a CRP taking after a regular meal sends your CRP up. So if we're checking labs and we didn't kind of ask a person about the last time they had a meal, it could be elevated from just eating. You're saying,
right? Exactly. And the literature has shown that this increased inflammation lasts a few hours.
okay.
And you know, a few hours later, usually it starts to stabilize around 5 to 6 hours. At that time, we're about to eat another meal. So again, we get another spike in inflammation. So it's a constant chronic process of low-grade inflammation that occurs.
Now, let me ask this just because I've heard before that food in general creates some form of inflammation in the body. Is that seen also with lab work?
Say that first part again. What was it?
All foods. So fruits, vegetables, meats, grains create some form of inflammation in the body.
Based on what we see, that is not the case. You can have you will not see an inflammatory response with plant-based foods for the most part.
Okay.
Yeah, you won't see that inflammatory response.
So, Robbie, if you have plant-based foods, and see, we need to do this study. We're going to probably have information on this with this next study we do. But let's say I have let's say I have a piece of okra. I have an okra meal, and okra meal A is raw okra. Okra meal B is steamed okra, and okra meal C is fried okra. Do you think okra meal C will raise inflammatory markers, whereas okra meal A will not?
With assuming there's going to be a hyperlipemic, so there's going to be increased fat in the blood. So I would assume that there would be some increased inflammation if there's much more fat in the blood, especially.
And let me put it this way: why don't we air fry okra C so we're not putting in oil, so we control for that?
It has never been done. I suspect that you wouldn't see a huge spike, but I don't know. Obviously, it has not been done.
Okay, we'll make a note. That's a future study.
Right.
Well, I'm gonna but I'm glad you showed the slide. I'm gonna invite Dr. Pam Atkins in. Dr. Atkins, how are you doing today?
Hi, good evening. Sorry I'm late. I'm doing well. Thank you. A lot going on in the world today.
I understand. Yeah, we're trying to deal with this cold and snow and broken water pipes and stuff. But anyway, so this is great. So we do animal protein. A single meal throws inflammatory markers up within 2 hours, and some of these graphs, those vectors are still in the upward direction. I mean, you got one on the third one is kind of starting to go down, but I can conceive that maybe the third or fourth hour you may still have a further increase. So by the time they eat the next meal, you're ready for another bolus. So this is quite interesting.
Right. It's interesting because when you eat, for example, there's research on plant food consumption and looking at artery function, and you'll see a peak in increased artery function around 4 hours, and you also see a peak detriment in artery function around that same time when you eat a highly processed, like for example, McDonald's meal. They've done this research where you eat a McDonald's meal, and at 3 to 4 hours, you'll see a huge plummet in endothelial function, which starts to come back to baseline around 5 to 6. And you'll see this with food, with plant-based foods. You'll see a huge improvement in endothelial function or vascular function around that same time, 3 to 4 hours, and then it comes back down to baseline levels. So you get it in both directions.
So, and Robbie, Catherine Welch likes the question about inflammation based on food prep. I want us to put a pin in that because later on in our discussion, I want to talk about that a little bit based on some of the work that we're doing and some of the work that's being done otherwise. But go ahead with your slides. That's great.
Sure. So getting more into inflammation, we're looking at atherosclerosis here. Atherosclerosis, like any other chronic disease, is an inflammatory process. I don't want to get too much into the biochemistry because it's complicated, but here's just an overview. There's various receptors on the cell, such as TLR4, which is a toll-like receptor, and TNFR here is the TNF-alpha receptor. So TLR4 can bind to a variety of things. LPS, which is after eating an animal-based meal, you actually find LPS in your bloodstream, and I'll show that shortly. Oxidized LDL, eating, there's been research on egg consumption. You'll find increased oxidized LDL. LDL is more likely to become oxidized after consuming eggs. And palmitate, which is a type of saturated fat that's found primarily in animal-based foods. And it's interesting because high-fat animal-based diets particularly, will increase palmitate concentrations in your bloodstream, which activates this receptor. And that recruits, you'll see here, these little blue dots are cytokines. So that and inflammatory chemokines, which attract macrophages, and they can innervate the endothelial cells and get in the layer under, get right underneath the endothelial cells and form fatty plaques.
And Robbie, I mean, this is a beautiful diagram, and I love the way you go through the details, because even though it's some of the details are beyond what the lay public will look at, but I think it's I like going into those details and then stepping back and squinting, and what I'd like to convey to the audience. If you look at this top diagram with this vessel, so this is the inside of a blood vessel we cut open, and you got these little cells here. We call them endothelial cells, and right where this little hump in the cell line with this yellow glob there, that's like a pimple. And anybody who's had acne had these little juicy pimples. And these juicy pimples, like if you sort of brush up against it, it'll pop. And so a juicy pimple is all an unstable plaque is like a juicy pimple on the skin. And it doesn't take much for the lining over this plaque here, which is analogous to an abscess, to rupture and open. And when this opens, you get these chemicals exposed to the blood flowing, and that triggers a cascade of biochemical events that ultimately forms clot. And so that can lead to a blood clot obstruction. So that's kind of an extreme case where you have a rupture of this plaque and you can get a clot formation. That's the mechanism of a heart attack. And what Rami is pointing out in such detail is that these are complex mechanisms by which how our immune system work. And if things go awry, you get false signals that bring in all these inflammatory cells that can further destabilize this plaque. And we're going to look at certain things that a plant-based diet affect. And so that's why we need to look at these biochemical precursors. But this is a beautiful slide here. I really like the details that you go through there, Rami. Right.
The inflammatory process, it's a maladaptive response when there's chronic inflammation. Your blood vessels are not used to having this chronic inflammation on an ongoing basis. You know, infections are usually, I got ahead of myself there. Inflammation is usually due to infections or some sort of injury, physical trauma. But when it's from other things such as diet, your body not really good at handling that.
Yep. And it's sort of like a ticking time bomb, because you have this underlying chronic inflammatory process. And this is an ongoing process. And what happens is if you get a trigger, like an acute infection, something that kind of throws this, beyond some trigger point, that's when you get plaque rupture, heart attacks, and various other things. And so that's when acute events occur. And so, but this is beautiful. Go ahead, Rami, take over.
All right. So I mentioned LPS is one of these things that can actually activate TLR4. If you'll notice, they had subjects consume either water, cream, glucose, so sugary drink, or orange juice. And you can see the endotoxin concentration. This is LPS. You can see over the course of 0 to 5 hours an increase in LPS in the bloodstream. And then on the right here, you can see changes in TLR4 gene expression in various white blood cells in the bloodstream. You can see how TLR4 gene expression with a cream is significantly higher than the other groups. So you can see that essentially after eating just cream, you're going to have LPS in your bloodstream, and TLR4 is going to be activated because of this.
And this is what, ice cream or yogurt?
No, it was just sugar-free cream, just for coffee creamer.
Boy. Celeste, you want to ask something?
No, I was going to say, is that we're talking about dairy when he says cream, but you answer the question.
Okay. Great.
And this is regarding LDL oxidation with 2 eggs per day. I mentioned that oxidized LDL is another thing that activates TLR4. You can see at baseline, they took LDL from subjects after 3 weeks of egg consumption, 2 eggs per day, and they assessed how its propensity to become oxidized. And what they saw is that its propensity to be oxidized was 34% greater after 2 eggs per day for 3 weeks. And they saw a lot more oxidized proteins as well in the cholesterol.
And share with the audience the oxidation versus antioxidants. I mean, most people understand antioxidants. You talk about an oxidized LDL. Put that in the context of what people understand regarding antioxidants and oxidation.
Right. So oxidation is essentially a molecular change in a molecule where an oxygen atom is usually added to a molecule, and this changes how it functions and how it's perceived in the body. Essentially, every process in the body, every single process is chemistry. So this simple oxidation, this addition of an oxygen, in a variety of enzymes, and even in cholesterol, your body handles it differently.
Gotcha.
Yeah, and antioxidants essentially neutralize the. You'll have reactive oxygen species such as free oxygens, and I'll show that later, that want to bind to different enzymes and different proteins and modify it. And antioxidants neutralize these reactive oxygen species before they can do that.
And by just adding an electron, right?
Yeah, they will add an electron, and it depends. There's different oxidants. That's one of them. Hydrogen peroxide, which is H2O2, it'll just essentially rob it of the oxygen and making it water. So there's different mechanisms.
Gotcha. Okay, great. We're going to become biochemistry experts tonight. This is great.
So I mentioned palmitate as a TLR4 binder, and this is looking at free fatty acids in the blood. So normal people, they'll have 0.1 to 0.5 millimolar of fatty acids, but you can see with low carbohydrate this increases, and with obese subjects this increases even more, and insulin resistant subjects it's around the same, and this can actually increase TLR4 expression. There's a lot of in vitro work where you simply provide palmitate to cells, and TLR4 is significantly upregulated, as is inflammation.
So explain the numbers to the audience. So the greater the negative number in terms of plasma-free fatty acids.
Yeah, these aren't, let me see here, pointer options pen. So this is not, these aren't negative numbers. These are just rough estimates.
Oh, I'm sorry. Oh, it's approximation. My bad.
Yeah, these are approximations, right. Right.
Yeah.
So
Okay.
fasting,
So the higher number.
free fatty acids are around 0.1. Fasting, it increases to up to 0.5. But if you're eating a high fat diet, it increases to 0.8. And if your insulin resistance is around 0.6, and obese 0.6 to 0.8. So this is a lot of increased fatty acids in your blood, which means a lot more palmitate in your blood.
Gotcha. So,
activate TLR4.
okay. So baseline should be 0.4 to 0.5, and it goes up on the. Okay, that's
right, exactly.
Yep. Impressive.
So there's all these different receptors: toll-like receptor, TNF alpha receptor, Ang2 receptor, and essentially there's different enzymes. So MAPK, NF-kappa B, NFKB, these are inflammatory proteins. And NOx is an oxidative stress producing protein. You can see here it produces O2 minus, which is a reactive oxygen species, and H2O2, which is also a reactive oxygen species. And this is what oxidative stress is. These two compounds are major sources of oxidative stress. So when you hear about oxidative stress, these are the compounds that are doing it.
Gotcha.
And they can actually activate these inflammatory proteins, and the inflammatory proteins can activate NOx. So there's a lot of crosstalk between these enzymes.
Wow. I need to get a copy of these slides when we're done.
So inflammation can lead to a variety of things: death signaling in the case when there's excessive amounts, a lot of cellular damage, because I mentioned that these oxidants can modify different proteins and destroy cell membranes. So it causes cell damage. It can damage DNA, and this leads to chronic disease.
Gotcha.
This is essentially the underlying cause of all chronic disease. You'll see increased oxidative stress, increased inflammation, and these are the primary pathways that do it. If you look at literature in every chronic disease state, this is the main thing that's causing it.
Wow. Okay.
So delving a little into heart failure, just to illustrate how important these proteins are. If you look at the left, you'll have a normal heart, and the right, with heart failure, you'll have significantly enlarged left ventricles, which prevents blood from being pumped out of the heart adequately. And I'm sure Dr. Montgomery sees these patients all the time. They have a lot of chest pain, and it's usually not considered reversible. Medications usually handle it, but we've demonstrated with diet, as we'll show later on, that it can be reversed. It's thickening of the heart wall and improvement in blood flow.
Yep. And a thickened heart, the squeezing is okay, but relaxation is impaired due to a lot of things. And if it's diabetics, you got infiltration of scarring, which is inflammation, which is what you pointed out. It could be due to ischemia, which is poor circulation of the heart muscle, because blood flow is needed to allow the muscles to relax. So the muscle don't relax. And if you can compare the left one to the right one, the chamber on the right is much smaller. So you got less volume going into that ventricle because that muscle is thick and stiff. So it doesn't feel as much. So if you had like a liter of fluid being pumped out of the first one and only 500 liters out of the second one, that's a 2-to-1 ratio in volume, and flow is volume. And so as Rami's pointing out, this thick, stiff ventricle can rob you of the amount of flow. The heart has to beat twice as fast and uses twice the amount of energy with less circulation.
Right. And so I mentioned that different proteins. So TLR4 here, if you look at people with heart failure with a reduced ejection fraction, so ejection fraction is essentially the ability of this heart to pump out blood, the percent of blood that leaves the left ventricle. You can see here that subjects with reduced ejection fraction have much higher TLR4 expression in their hearts. And they also have much higher NOx expression. Remember, NOx produces oxidative stress. So you can see here that much higher TLR4 inflammation and oxidative stress proteins. And if we look at, they actually took heart sections and stained it with fluorescent staining. So the more green you see, the more TLR4 you see. So you can see in the myocardium slices, much more TLR4 expression in the hearts of those with heart failure with a reduced ejection fraction.
So green is bad here.
Green is bad. Green means more TLR4 activation. So essentially, this is what is causing heart failure, is TLR4 activation.
Wow.
as well as NOx. There's an interplay here.
And we're going to
And,
look at that in the next study. Okay.
Yeah, if you look at NOx activity, this is in failing hearts, diabetic cardiomyopathy, and non-failing hearts. You can see NOx activity is much higher. So this is just showing, just kind of trying to bring it all together, showing that TLR4, inflammatory signaling, NOx, oxidative stress is all increased in the failing heart. And these are human hearts.
Wow.
So animal models and research, we do a lot of animal research. I know there's a lot in the people in the plant-based or vegan community that are against animal research, and I sympathize with that. But to understand mechanisms, it's almost impossible to study mechanisms in humans in the same way. Like those hearts were isolated from surgical intervention to do coronary artery bypass. So you can't really do an intervention and take people's hearts that are, it's not ethical. So animal research is really one of the only ways to do that.
Either sacrifice the mice or sacrifice the humans, huh?
Right. So this is an interesting study, actually. So they had wild type mice, and they performed a surgery which induces heart failure. So they ligate, they close off the coronary artery, and it causes reduced blood flow to the heart. So it mimics atherosclerosis, which reduces blood supply to the heart. That's essentially what they did to these animals. So they had a wild type group, and they also had a NOx knockout where they genetically modified these mice to have reduced expression of NOx. So NOx, again, produces reactive oxygen species, oxidative stress. What they find is very interesting. So this is looking at the amount of superoxide is oxidative stress. And you can see here, sham means they didn't do a surgery. It's just a control group. MI is myocardial infarct. You can see a lot greater superoxide production. But when they took out the NOx protein from these animals, you can see much, much less superoxide production. And when they looked at the animal ejection fraction, you can see ejection fraction was preserved with NOx knockout, whereas diminished with the wild type animal. So essentially what we're showing is that when you eliminate NOx, you get a preservation of ejection fraction. So the heart is protected because you have much less oxidative stress. And if you look at the weight of the heart, left ventricle thickening, as we saw with that picture of the heart, the heart wall gets very thick, and you can see here when you took out NOx, the weight was very similar to control. So, and there's a huge increase in heart weight when you in the wild type animals.
So basically, the heart was essentially, when you remove that NOx, which is again a major mechanism of oxidative stress.
Right.
If we genetically remove that major contributor of oxidative stress, the hearts of the mice tolerated the heart failure better. So if you remove that mechanism of oxidative stress and then give a heart attack, compared to not removing it, the hearts with the removed oxidative stress mechanism were stronger and smaller and more compliant than the hearts that did not have the oxidative stress mechanism removed. So again, what you're showing here is a mechanism of oxidative stress. So despite an injury, it's the oxidative stress that happens after the injury. So you can argue that if somebody was on a nutritional program of some type, so if you can remove that oxidative stress mechanism by some other means other than genetically, you could probably have a similar outcome. Is that a reasonable?
Exactly, and that's exactly what I'm going to show as well with plant-based constituents, how they can actually reduce NOx expression.
So how was NOx knocked out? How did you take NOx out?
So
how did you eliminate NOx?
They do it. They can do it in the cardiovascular specific NOx knockout, where they can do silencing RNA, or they can just where they essentially inject in the heart an RNA which prevents NOx synthesis from happening.
I see. Okay.
That's one mechanism. I have not done genetic knockout like myself. This is very advanced research. I'm not a geneticist, but that's one mechanism. And there's other ways to do it as well, where you can modify the DNA in other manners.
Fascinating.
Yeah. This last one is looking at fibrosis. You know, it's a lot of collagen buildup. And you can see with the lot of fibrosis with a wild type animal compared to the NOx knockout. So this is just showing in every way, essentially, the heart physiologically is preserved in a much to much greater extent with reducing NOx expression.
Fascinating.
So,
and fibrosis for the audience is scarring. So less scarring is seen after a heart attack, which is a major thing that we deal with.
Right. That's what. So the red up here, this is staining. This red, this innervations here, this is all collagen. You can see much less red with a NOx knockout. So I've gone over how NOx produces these reactive oxygen species, which activates these inflammatory proteins. So how does our body combat that? So there's two major proteins that do this. NRF2, which is, if you look in the literature, it's a master antioxidant enzyme regulator. It produces various enzymes, NQO1, HO1, GSH. We won't go into those details. And the other protein is SIRT1, SIRT2 and it produces other antioxidant enzymes. And these are known as detoxifying enzymes because they can eliminate compounds found from broken up proteins that are damaging to the cell, such as iron. Free iron is very damaging. So heme oxygenase can clean up that iron that's flowing around. These antioxidant enzymes can also neutralize reactive oxygen species, preventing the activation of inflammation. And SIRT2 can directly inhibit inflammation. So NRF2 and SIRT1 are master regulators of antioxidant enzymes, which leads to the decrease in oxidative stress inflammation. This is what I think plant-based diets are acting through, is NRF2 and SIRT2 and I'll show some of that evidence. So I think that these enzymes are key in being protective.
Nicole is jumping to the punchline. Whole food plant-based is the way to go. Well,
well, we have to prove it first. Nobody's looked at these enzymes with a plant-based diet, but that's what I hypothesize is happening.
That's right.
And I hope that some of our research we can show some of that.
So hold on, Nicole. Wait till we get our study done.
Yeah. When you have heart failure, though, all these pathways are inhibited. So you have to have some mechanism to increase the expression of NRF2 and SIRT1, because then you won't get the reduction in ROS and inflammation. So what do plants have that animals don't? So this is the main thing are phytochemicals, which by the name phyto is plant, so plant-based chemicals. And the main thing that we study in our lab are polyphenols. And there's a variety of plant polyphenols, very rich in berries, greens. Essentially every plant has polyphenols in various amounts, concentrations, and very different types. And they all have their own independent effects. And this is what I think is the main protective component of plants. And this is what elicits the effects that we see clinically are polyphenols. That's why I hypothesize.
Now, the polyphenols have an antioxidant mechanism effect, or
they have antioxidant effects. They can bind to different inflammatory proteins themselves.
Okay.
They have essentially every step of inflammation, oxidative stress. They're involved. They actually can enter the cell and target these enzymes on their own, and they also can scavenge ROS on themselves. But that's more of a superficial effect. They have much more potent cellular effects in the cell.
Now, when we talked about this before, you shared with me that these polyphenols are very short-lived. Is that just short-lived in the plasma, or are they acting more in a prolonged way intracellularly?
Yeah, so there's parent compounds here, and actually you'll find polyphenols in your bloodstream after a meal for up to 48 hours because there's all these metabolites that are produced from your gut microbiome. They'll break these down. Your liver will further modify them. So you can eat a parent, like this flavonoid here, you can eat this parent compound, but it won't appear in your bloodstream. You'll get all these secondary metabolites from microbial degradation in your large intestine and your liver modifying in a different way. So you'll still find polyphenols in your bloodstream even 48 hours later.
So basically, the metabolites are active and are playing a role as well. And you
Exactly.
you mentioned an interaction with the microbiome, which we've talked about in other shows here, and it starts to get pretty complex in an interesting way.
Yes, definitely. Yeah, a lot of people hypothesize that the effects of polyphenols, beneficial effects, are not from these compounds that are found in the plant intact, but actually their degradation and their metabolism, all those metabolites are what's actually acting in our body, and that's what would be exerting those beneficial effects.
So if your microbiome is not intact, then it's possible that you are not getting adequate metabolism of the polyphenols, and so you may not be getting full benefit of the polyphenols.
Right. Yeah. Your microbial, if you have poor gut microbiome, not the right types, you can actually not synthesize some of these compounds. But even consuming, there's been studies where they just give people polyphenols and their microbiome shifts. So obviously, plants are going to be the way to do that. So
fascinating.
Yeah, it happens within just a couple of days too.
So, Robbie, in this part one section, wrap it up for our group. How do you put? I know you may have a few other slides, but as you go through them, put this picture together for us. This has been a fascinating, intricate dissection of inflammation.
Right. So, essentially, oxidative stress, inflammation, are the main drivers of chronic disease, all chronic diseases, and if we can inhibit these pathways, as we probably can with diet, then that's how essentially plant-based diets function. That's how they're protective, is by inhibiting oxidative stress inflammation at the cellular level.
Wow. So if we can do knock the knocks like they did genetically, if we can knock the knocks with our diet, then we can get some of those benefits that you showed in that animal model.
Right. And I'll show some of that research as well in the next part.
Wonderful, wonderful. So is this a good time to take a break, Rami? Is this good on this part of your presentation? Because what we're going to do next is talk about some of the physiological effects, such as blood pressure and circulation. So we've wrapped up the whole concept of inflammation, and I want you to stay tuned, because on the next section, we'll talk about blood pressure and circulation. So we're getting to some of the physiological effects. And in our section 3, we're going to put it all together. So you all hang with us. I know it's been a little bit tight and detailed, but Rami's breaking it down because I want you to understand these details. So hang tight for the next section. Okay, we are back, and thank you for being with us. We will go to part 2, because now I'm going to talk about blood pressure and circulation. And so what we're doing is walking through the process of the mechanism of what we eat and how it affects our system from the cellular level to the tissue level to the whole body system level. So the first section, we dealt with inflammation. In summary, inflammation and oxidative stress, these are biochemical processes that occur at the cellular level and the tissue level. Rami showed how you can have a blood vessel that can have chronic inflammation, where the lining of the blood vessel can be damaged due to that chronic inflammation and can set it up for rupture or damage. That could lead to mechanisms of heart attack. He showed you how intracellularly, how the different mechanisms of oxidative stress that can lead to thickening and scarring of the heart. If you remove that mechanism of oxidative stress, you remove the effects. Even in the setting of a heart attack, the body can recover in a way, in a less damaging way than if you have ongoing oxidative stress. And so he showed you an example with a genetic way of removing, but there are ways you can remove it from a lifestyle standpoint, by removing these mechanisms of oxidative stress, we can help preserve our tissue and suppress chronic illness. So without further ado, I'm going to bring Rami back, and we're going to go to part 2. And we lost Dr. Palmer. She's been having some power issues throughout the day. So I'll check in on it as later in the presentation. But so part 2, we're dealing with blood pressure and circulation. And I just want to share this with the audience that now we're going to look at mechanisms in terms of how it affects hemodynamics, things like blood pressure and circulation. So anyway, Rami, go ahead and let me bring your slide back up. There we go.
Can you see me?
So where are we with blood pressure and circulation? What are some of the biochemical reactions that affect circulation of blood pressure?
So let me see here. So before we touch into that, I just want to share just a couple, if that's okay, Dr. Montgomery.
Sure.
Of how these polyphenols can target, because this will be important when we actually get into the discussion about blood pressure.
Mm-hmm.
So, because blood pressure, hypertension, it's also an inflammatory and oxidative stress process. So I mentioned that NRF2 is a key antioxidant enzyme, and when you essentially cause heart failure in mice, and then also provide polyphenols from berries, you can actually see a huge spike in NRF2 expression, which is an antioxidant enzyme.
So I'm sorry, go back to this other one. The sham here is walk through this slide, because this is a gel, and so you have your gel here showing that you have increased NRF2. Explain this to the audience.
Right. So these blots, this is the amount of protein found in the heart of NRF2. So the greater the density it is, so the more protein expression. You can see how this is a huge splotch of NRF2. So that means that it's highly expressed in the heart, in the nucleus. This is just a loading control to show that it was done correctly. And you can see here, this is graphically demonstrating what this is. So
IR stands for what again?
Sorry?
IR is what? Inflammatory ischemia reperfusion?
IR is ischemia reperfusion injury.
Okay, okay. So that's like your fake heart attack or whatever.
Right.
Okay. And then so basically and just for the audience, ischemia reperfusion, you close the artery for a period of time and you suffocate the tissue, and then you open it up. So that the ischemia is when you close the artery and then you let the tissue, sort of like when they put that tourniquet on your arm and your arm turns blue, and then they take the tourniquet off and you get more blood flow. So you get that ischemia, low blood flow, and then you let it reperfuse. So the sham is a fake. You always have to have a fake, because you know, some of the rats may think these are human. The rats. So you got to have the fake where it's kind of a blind. It's a true placebo. Then you got this group, and then you add your berry. This is urethylene blue. This is the compound from the berry, right?
Is a metabolite found after consumption of berries. You can see it in your bloodstream.
Gotcha. And so that allows more NRF2 formation despite this injury or ischemia and reperfusion mechanism.
Right. An antioxidant enzyme, exactly.
Yeah.
And this is in a model of heart failure as well. It's SIR2-1. This is with curcumin, which is a turmeric polyphenol. So consuming turmeric here, you can see that SIR2-1 is decreased with the heart failure animal, but increased with addition of curcumin. So both of these proteins are affected by dietary interventions.
Wow, and that's interesting. It's interesting because you see the sham where there's nothing happening. Curcumin plus sham is essentially the same. It doesn't really throw it up even higher, but it comes into play when you have an injury. So so it's fighting against inflammation. So it's sort of like there's no fire to put out here with the sham, but there is a fire to put out here with the MI.
Right.
Fascinating.
And this is just looking at fibrosis, essentially just showing very similar effects. You can see much more fibrosis. All this blue is collagen scarring. So all this blue is much more scarring, and then it's diminished with the addition of the curcumin.
Yeah, we use curcumin in our clinical setting on a regular basis. We may need to just start using it regularly with our patient with ischemic heart disease.
Yeah, it's very potent.
Wow.
So this is a defined plant-based diet, inflammatory markers, and oxidative stress. This is actually from the study Dr. Montgomery and I co-authored. You can see baseline after 4 weeks of consuming a defined plant-based diet, very rich in raw fruits, vegetables, seeds, dramatic decrease in CRP, which is inflammatory protein, IL-6, 23% reduction. Myeloperoxidase is a oxidative stress marker. It almost reached significance, but a 23% reduction. And then these are white blood cells, which are elevated in when there's inflammation. You can see how they're all decreased after the plant-based diet or dietary intervention.
And this is important because this is only a nutritional intervention, and these were people with high blood pressure, hyperlipidemia, and they consumed the plant-based diet. They went from a standard Western diet to a plant-based diet, and you saw these very significant abnormalities. And if you look, certainly in the white blood cell count, that 6.3, that's within the normal range. And so lots of people walk around with white blood. The range of normal white blood counts like 3 to 10, and or 10.5 or whatever it is. And this was in the middle of that range, but it came down very significantly, as did all the other parameters. I can't remember, Robbie, this myeloperoxidase. This was in the normal range also, was it?
I don't recall, but a decrease nonetheless demonstrates that there's reduced oxidative stress.
Yeah. And see, the thing is that the reason I'm pointing out that these things were in the normal range, the CRP was elevated, but the reason I'm pointing out they were normal range because it's harder to show a significant decrease in something that's coming from a normal range than something that's elevated. So these patients were not in any acute stress, but probably had, say, a low-level smoldering inflammatory process that was decreased, which is important because it's if we had people who were having heart attacks or unstable angina and they had increase in these inflammatory, increased white count, increased myeloperoxidase, then you can probably more easily show a decrease there because you're putting a fire out. But here you show a significant reduction even in the so-called normal setting. So I think that's an important thing to point out. I say normal setting, it's in the stable setting. It's probably not normal, but anyway, not biologically normal. But anyway, go ahead.
Right. This is in hypertensive animals here. So I'm just trying to demonstrate here why whole foods are key and you can't just take a pill. So here's gallic acid, which is found in abundance of plant foods. And these are hypertensive animals here. And this is the amount of NOx expression. There's different isoforms, NOx 1, 2, and 4. You can see gallic acid was particularly potent against NOx 2. You can see an increase. SHR is spontaneously hypertensive rats. Huge increase in NOx 2. And then gallic acid resulted in a decrease in NOx 2. Didn't really affect these other ones too much. In a diabetic model, you can see this is a different polyphenol, ellagic acid, also found in abundant of different fruits, pomegranate, berries, nuts as well. You can see NOx 4 is inhibited, whereas NOx 2 is not changed. You can see decrease in NOx 4 expression. This is in a diabetes model with hyperglycemia. You can see hyperglycemia caused an increase in NOx 4, but ellagic acid decreased. So all these different polyphenols combined in a diet could potentially affect all different types of NOxes. And it's a cumulative effect that is likely the main benefit to a plant-based diet. You can't just take a pill, a single polyphenol, and expect to be protected.
And it's probably an interaction between those polyphenols. And as you alluded to, the microbiome is interacting with these polyphenols. And so there could be polyphenol to polyphenol interaction as well as polyphenol to cellular mechanism and microbiome. Right. So
definitely synergistic effects.
Yep.
So now let's delve into hypertension a little bit. So, what are the medications, Dr. Montgomery, that you prescribe for blood pressure?
Well, you're alluding to it right here with the angiotensin 2, so we can block
Right.
the angiotensin 2 receptors.
Right. Yeah, angiotensin receptor blockers and ACE inhibitors. So
Yeah.
ARBs and ACE, right? So
And that whole cascade, the ACEs and ARBs.
Right.
And of course, you got calcium channel blockers, which affect smooth muscles, which is part of that diagram you're showing. Of course, beta receptors are at the cardiac set, and also peripheral beta 2 receptors.
Right. Yeah, so in the case when there's increased angiotensin 2 in the blood, this can actually drive hypertension. This is a cross section of artery. Here are the vascular smooth muscle cells. Here are the endothelial cells. Increased Ang2 can result in a constriction of the blood vessel, which results in increased blood pressure. If you're squeezing, a bottle of water, there's going to be a lot more pressure in that bottle of water. So that's essentially a similar mechanism here.
And just by way of context for the audience, these molecules have an important role because the body, there's a situation where you have to vasoconstrict certain arteries. So vasoconstriction isn't a bad thing. For instance, let's say if you're in an auto accident, let's say a healthy person in an auto accident and you're bleeding out, you need to preserve circulation and direct circulation to areas that are important. So you need to circulate blood to the brain or to the heart, and you may circulate blood away from the kidney or away from the GI tract. So you may need these hormones to vasoconstrict in certain areas.
Right.
The problem we have is that if in a pathological state you have an excess expression of this, then you're vasoconstricting everywhere, and that's when you cause a mess.
Right. So the main cause of this is, Ang2 synthesis. ACE inhibitors essentially target ACE right here. Mm-hmm. And there's different enzymes that produce Ang2. And essentially inflammation can drive this process. This is known as RAS, renin-angiotensin system. So inflammation drives Ang2 production. Inflammation in the liver and the kidneys drive this process. And it's interesting because Ang2 is also produced in almost all tissue, including the heart and blood vessels as well. So you can have Ang2 synthesis in all tissues. And when this happens, it can respond to the angiotensin receptor. That's what AT1 are. So you mentioned angiotensin receptor blockers as a medication. That's what this is blocking. It's blocking this angiotensin receptor right here, like losartan. This targets this receptor. And if Ang binds to these receptors in endothelial cells, it can activate NOx, reducing nitric oxide, which is a vasodilator. In the vascular muscle cell, it activates NOx, which can impair calcium handling. Calcium is how the smooth muscle cell, if there's too much calcium, it constricts. So essentially, you get increased vasoconstriction, which results in hypertension.
And in addition to at the tissue level, you activate fibroblasts. And so certainly in the heart, you get scarring as well.
Definitely. It affects all tissue. It affects, Ang can bind to, in our model, in the lab, we're actually doing an angiotensin 2 model in rats, and it actually causes hypertrophy of the heart just from infusing Ang2. And this could be due to elevated blood pressure, but in cardiomyocytes that we're growing, human cardiomyocytes, when you give them Ang2, they express NOx and express inflammation. So independent of blood pressure, Ang2 is having this detrimental effect.
Yeah, I know it's an independent factor because I did studies back when I was a fellow. I show in African American individuals, we have a higher expression of renin-angiotensin both at the tissue level and also at the serum level. And so we looked at a DD genotype, and so we have a double deletion of that, and so we have a higher renin-angiotensin expression. And what we know from African American patients, there's a greater amount of left ventricular hypertrophy for a given amount of hypertension compared to non-African American, non-Black patients. And if you have Blacks who are from the African continent who have never intermarried, they have an even higher amount of renin-angiotensin activity. So we know based on genetic studies across population lines that this as an independent contributor to hypertrophy, as you pointed out, and then of course fibrosis as well. So and it may be really the mechanism in most people, and the elevated blood pressure may contribute a little bit, but the elevated blood pressure is probably a sign of what's going on here.
Right, definitely. And as I mentioned, Ang2 can be synthesized in all tissue. You can see ACE is expressed in almost every single organ here, which is angiotensin converting enzyme. ACE and AT1R, in my view, are the main targets of polyphenols. And
I'm not going to get you to comment on coronavirus because that's not your area, but the mechanism by which the coronavirus gets in is the ACE2 receptor. And so the amount of receptor activation in our tissues, particularly the lungs, but also on the heart and other tissue, the GI tract, is how this virus gets into the tissue and contribute to this. So again, this mechanism that we're dealing with can be associated with that. And we know that diabetics have upregulation of this ACE2 receptor as well, but also people who are on ACE inhibitors, because we're treating hypertension, may have an upregulation of these receptors. And this is part of the mechanism of how people with these chronic illnesses are predisposed to this illness, in addition to suppressed immune system. But anyway, I wanted to throw that out to give some context.
That is interesting. Decreasing ACE might have an impact then with corona.
Yeah. But the thing is, as a natural downregulation is the mechanism that you want to go for.
So speaking of ACE here, so this is, and again, hypertensive animals. What they provided is gallic acid, which is found in essentially all fruits. And you can see here, ACE expression is significantly upregulated in these hypertensive animals compared to control. And gallic acid supplementation decreased its expression. You can see the density of these bands is much less. So there's much less ACE expression. So this is exactly what an ACE inhibitor would do, would decrease ACE. Like losartan decreases angiotensin receptor. This is the angiotensin receptor. You can see gallic acid is decreasing the expression compared to the hypertensive animal here. So these polyphenols are acting like drugs. They're acting like ACE and ARBs in this animal model.
And it's an interesting point you make, because the fact that they act like drugs, we think of drugs, and they are chemical molecules. But what I want, and what you're doing such a great job, is expressing the complexity of the foods, because they have a biochemical effect. And so, we talk about food as medicine, and the reason I want to go through these mechanisms is because I wanted people to get a sense of how food is medicine, because these mechanisms have a direct effect on cell receptors and the like, and it has an effect similar to medications, but similar is not the right word, because it has a more complete effect, because it's more comprehensive. Medication may affect one receptor, but these foods affect not only one receptor, but it affects a cascade of receptors in a synergistic fashion, without side effects, of course.
Right. It's the power of plants here. And if you look at our own study that we co-authored, Dr. Montgomery and I, you can see baseline, these are hypertensive subjects, starting out systolic blood pressure of 146, and by week 4 on the plant-based diet, down to 130. And if you notice the stark change here in the blood pressure medications, so the decreased blood pressure medications and decreased blood pressure. So the diet was more efficacious than the drugs. If we kept them on this 1.6 medications on average, blood pressure would probably be a lot lower, but they no longer needed it. They would become hypotensive.
And that's right. We frequently have to wean the medications. That's why you see week 1 through 4, we see them weekly, because medications can drop very significantly, even within the first week. As you see it here, you got 15 points almost on the systolic pressure. and 10 points in the diastolic pressure in the first week alone. And so it has a very, very impactful effect on blood pressure. And at the same time, we saw a 40% reduction in overall medications, even greater here with just blood pressure medications.
I think this is a wonderful point. You know, when patients say, how can I reduce or get off of blood pressure medications? The more that they eat these high antioxidants, colorful vegetables and fruits on a plant-based diet, then they can have more power over improving their blood pressure and perhaps decreasing the need for medication.
Yeah, that's exactly right. And, you know, we did this 4-week study purposely to show how the actual food could be prescribed in a way and have an immediate effect. I mean, someone walks in an office and, you know, the blood pressure 160 over, 95 or 100, and they're on no medications. They say, well, you know, do I have to start a medication? So, well, you don't have to. This evidence shows how food could be prescribed right away, and we can get effects in the first week or two. So, it's oftentimes people think of lifestyle changes where you eat, a piece of broccoli today, and about 10 years, something good will happen. But that's not necessarily true. You have an effect here that works on your hemodynamics right away. I want to let the audience know we are going to go over today. I didn't mention it early on, but I want to get these concepts in. So go on, Rami. Just we'll keep moving along.
Sure. This is the plant-based diet on heart failure here. This is a case series with 3 subjects. You can see that ejection fraction increased, which is the percent of blood pumps out of the heart. LV mass decreased, so the heart was becoming less thick, because the thickening of the heart is what prevents that blood from flowing out. Increased stroke volume, cardiac output. This is only in 3 subjects, and you can see here with the coronary angiogram. If you want to describe this, Dr. Montgomery.
Yeah. So in the coronary, in the before, you see this yellow circle here. And I suspect you can't see my arrow, but just look in the yellow circle. There's an arrow. There's a little pinched in area of this artery coming out, and the little black thing that goes up toward the edge is my catheter. It comes into the artery. So they have the left main, and that branch that comes out is what we call the left anterior descending. And at the very beginning of that branch that goes across the page and before, it's pinched in right there. And if you look at the after, that pinched in area is no longer there. So that pinched in area is the stenosis. We estimate about 90%. And even the branch before that, we call the left main, is smaller than the one before here. And so in about 5.5 months on just a plant-based diet, this artery opened up. And so, again, this is due to the reduction in inflammation. If you remember that very first slide that Rami showed, it showed the opening of an artery, and it showed that little plaque there, the little hump inside the lining. And that's where you see that pinched in area. And that was that completely or nearly completely regressed on the plant-based diet. So that was something that was demonstrated. And that's been then shown by other authors. Ornish and Esselstyn shown that, not necessarily to this extreme of the degree, but they have had vessels that visibly revascularized, and over the course of a year showed significant revascularization over a population of patients. And we actually also, in the ejection fraction, as Rami pointed out, this is a case series of 3. The reason we only used 3 is because we had cardiac MRIs before and after the nutritional intervention, which is hard to get the cardiac MRIs done logistically. But we see this routinely. We see improvement of the ejection fraction routinely with echo and so on. So I can probably get another, 50 to 100 patients over the years that we've seen improvement in ejection fraction. I didn't put those in this population because, echo before and after isn't quite as quantifiable as cardiac MRI. But we plan to do some studies with larger number of cardiac MRIs to demonstrate this, in fact. But the key thing is reduction in LV mass, improvement in ejection fraction, stroke volume, and cardiac output are all important findings that Rami demonstrated in the animal models, we show it here in human studies, or at least this case series, at least.
Right. And if, ejection fraction of less than 45%, Dr. Montgomery, correct, that's considered heart failure?
Yes. We kind of, the different stages, but certainly, less than 50% is definitely abnormal, but then, 45% or less with echo, you say you're beyond the error range. And so it did, but this population, it went from an average around EF of around 20% to around 44%. So even though it's still abnormal, this population here at baseline, if your ejection fraction is less than 35%, you need a defibrillator. You have increased risk of sudden death. So this population went from, even though it stayed abnormal, they went from an increased risk of sudden death on average to not being an increased risk of sudden death. So, and in one patient, this patient here had an EF of 24%, went up to 50%. So,
right. Let's show that.
Yeah, so you can see a real significant improvement function. And so, yeah, you're going to demonstrate her echo.
Before is with a 20 low 20% ejection fraction, and after is at 55% about. You can see.
Yeah, about 55. I think the imaging people read this around 24, and they read this at 50 on the after. But you can see it. Go ahead and recycle that again. You can see how this hardly thickens here. This is the left ventricle, and see how this squeezes in. It's pretty obvious there. And this is only on a nutritional intervention.
Right. You can tell how forcefully the heart is pumping blood on the right and how rhythmically it is. It seems there's some sort of arrhythmia on the left as well.
Great job.
Right. Well, that's all the major points I wanted to touch on regarding.
Okay, we're going to take a quick break, and then we're going to come back and put all of this together. We're going to take section 3, and we're going to talk about how we deal with chronic disease and put all of this together. So stay tuned. This. So as we move forward, Rami, come on back. Dr. Atkins, come on back. Let me bring your slides back up. So help us out here. So chronic illnesses, a lot of people say, okay, this is all well and fine. You can knock the knocks. You can, show these wonderful pathways. But, I've got, diabetes, and I've got arthritis, and so on and so forth. What does that mean to me in terms of, how we deal with chronic illnesses in general?
Right. Well, as I mentioned, these inflammatory pathways really are affect every single chronic disease state. So if you have increased inflammation, this causes insulin resistance. This can drive insulin resistance. And all these diseases are very linked. For example, if you have diabetes, it's actually an animal model to induce diabetes to cause heart failure.
Huh.
If you have heart failure, it can lead to COPD, and vice versa, actually. These are all very, the inner organ link between dysfunction of one organ leading to another is very common. And I'm sure you see that a lot.
And so essentially, and this slide does a good job of demonstrating that to a certain extent, but inflammation anywhere is essentially inflammation everywhere.
Right.
And so somebody that has vascular inflammation and vascular disease, at the same time, something is happening in the pancreas, something is happening in the muscle tissue to contribute to insulin resistance. There's hepatic or liver dysfunction that's going on at the same time.
Right. And I had mentioned, and we talked about angiotensin II driving hypertension and affecting various tissue. In our current animal model, where we infuse angiotensin II into rats for 4 weeks, we see that almost every tissue that we looked at, the heart, kidney, lungs, even the brain, is the brain surprisingly will have increased expression of oxidative stress, which drives hormone release of angiotensin II and adrenergic signaling, as you use beta blockers to try to prevent that. But every single tissue that we looked at is affected. So it's really, if it happens in the body systemically, it's affecting all tissue.
So we're really dealing with, and that's what I like for people to understand, because and you said the magic word is happening systemically, and let me kind of give people an understanding for the approach, because if someone has a heart attack or if someone has liver dysfunction, whatever the case is, in medicine we tend to treat that organ. We'll give you a medicine for the lung or whatever the case is. But think of it this way: if you have inflammation or increased oxidative stress, you have a systemic disease. It's analogous to having metastatic cancer. People understand cancer. The doctor said, well, has the cancer spread? And you say, well, the cancer has spread. It's all over the body. That means it's systemic. It can go to the brain, the lungs, the kidney, the wherever. Maybe it starts in the colon cancer, but it can metastasize. Well, chronic illness is sort of like a metastasized metabolic process. It's systemic. It's all over. So we may give you one medication that affects one biochemical pathway in one organ system, but we have to treat it systemically. So the power of plant-based foods isn't so much that, we can show that plant foods affect this biochemical process here, but it has a systemic reaction. And as you pointed out, there's an interaction like polyphenols. There's an interaction with the gut microbiome. But guess what? As you'll remember from our microbiome discussion, we know that plant foods enhances the microbiome. So if you're giving, say, an angiotensin receptor blocker, it's not going to enhance the microbiome. In fact, some of these drugs have an adverse effect on the microbiome. And so plant foods work synergistically. So on one hand, the plant foods are delivering polyphenols. On the other hand, the fiber in the plant foods feed the microbiome and making them happy. And they're doing stuff, and they're breaking down the polyphenols and creating these wonderful metabolites, which are then going all over the body, creating positive biochemical reactions. So there's a pleomorphic effect of these foods that works in a way beyond what we understand. What Rami went through is very complex, but this is a very tiny, you got a 0.0000001% level of understanding of what's really going on. So the complexity of these foods it's beautiful, as Dr. Colin Campbell likes to describe it, as a symphony, symphony playing beautiful music. So I like the way you point this out, and these mechanisms work all over. And so these chronic illnesses are pretty much the same illness.
Right. And it's surprising that this research in this plant-based field looking at these mechanisms and these pathways is essentially undone. This is a huge frontier of potential research that I hope we can investigate and look into, which is, obviously we have some plans with some patients with coronary artery disease, but it hasn't been looked at. And this would really get this mainstream if we have mechanisms tied with physiological effects.
Well, it'll certainly raise our understanding. Mainstream that's going to be a political process that's going to help. Dr. Atkins, what I'm talking about there, she's laughing. but it'll help us understand and give us more argument. But the mainstream, I don't know, we're going to have to, get some Army tanks or something. I'm not sure what that other process is going to take. but just to share with the audience what we have up our sleeves is we're looking at a plant-based diet compared to a Mediterranean diet or other diets, and we want to look at the relative biochemistry of those two nutritional interventions. And we're going to look at all of these detailed biochemical reactions and compare them and map these changes along with the change we see in standard biomarkers, as well as the change we see in clinical scenarios where we look at cardiac perfusion studies and look at, endopat measurements of vascular function. So it's really going to look at what we're going to try to do is map what's happening at the subcellular level and correlate that with what's happening at the clinical level. And try to map these changes in a way that we can better understand the mechanism. We're also going to try to look at a little bit of the microbiome as well in this population. So I'm really excited to look at this. Any final words, Rami?
What I had said previously regarding it being a frontier, this is we know plant-based diets work based on what we've seen, what you've seen clinically. Firsthand, I'm sure that's very powerful, more powerful than any study. But my goal is to get this mainstream, especially in medicine, and by researchers too. And I think it's getting there. But I think going into these mechanisms and showing researchers that, it's influencing the pathways we've been studying for decades. That is key to getting plant-based, getting physicians and other scientists on board with plant-based diets.
Yeah. And you mentioned mainstream. I think it'll give us more armamentarium to go and talk to my colleagues and say, look, guys, here's the science behind this, and you can't really refute this. And so, you can, right, but the evidence will speak for itself. Dr. Atkins, any final words on your part?
Well, I'm just excited with your research and your study, and it just confirms that what we see clinically, when we put people on a plant-based diet, high in antioxidants, a more raw, high enzyme, we see symptoms better. Joint pains decrease or go away. We see blood pressure, stabilize, improve. We see blood sugar. We see weight loss. Cholesterol comes down. Things fall into place. So it is a symphony we need. If we don't, so that can make a difference for patients that are willing to make changes in their diet and their lifestyle. It can be quite profound.
Wonderful. And with that, we'll let that be the final word. I'll see you guys backstage. Again, thank you very much for joining us tonight. We did go over, but these were very complex concepts we covered tonight. We wanted to really get this information across so you can get a grasp on it. We're going to deal with this topic over time. We'll probably break this down in some bite-sized discussions in the future. But I really do think this is important. It's important for you to understand why you're getting healthy. It's important for you to understand why it's important to eat these plant foods, a large variety of plant foods, not just to say, it's good for me, but to understand that, hey, this is something that's very important and it works by this mechanism. And not only that, to understand how the adverse effects of the other foods. He showed you data in terms of how consumption of animal protein increases inflammation in a very short period of time. So the more we understand these things, the more we understand the importance of making these lifestyle changes. So anyway, thanks again for joining us. And of course, always until next Monday. Next Monday, we'll be visiting with an Olympic athlete who made a transition in her lifestyle, and we'll share all the details with you. But until then, I want you to keep it fresh, natural, and live.